Uh, يعطيكم العافية. We'll continue the last lecture of the mid material of immunology. So before we get into this, let's do a small review about the T cell receptor that is bound in this case here to, زي ما حكينا, it's always bound to CD3. CD3 acts as a signal transmission conductor to conduct or transmit the signal from the T cell that has been occupied by its unique antigen, and then send the signal down to the nucleus. نحكي محاضرة قادمة إنه what happens in the nucleus briefly يعني that will turn on interleukin-2 production for activation of the CD4 cell and other cells. هلا CD4 acts as a stabilizer. مع مين؟ It will stabilize with MHC2 producing or carrying cells that are going to interact with this CD4 cell. هلا بدنا ندخل على this new concept. We touched on it briefly. حكينا إنه CD4 lymphocytes perform their regulatory function. حكينا إنه they can activate CD8 cells. This is the interleukin-2 pathway, the cellular pathway to make the cytotoxic activated cytotoxic T cells. حكينا the other pathway is to go towards the humoral pathway to activate B cells to become plasma cells that produce antibodies. They also حكينا they help macrophages affect their delayed hypersensitivity. هلا the first two functions اللي هي cellular function والhumoral function is carried out by different subpopulations of CD4 cells. إيش يعني؟ يعني في CD4 cells متخصصين في activation of CD8 cells ودائما بيحاولوا ينقلوا الجسم towards a cellular response. هدول they are called th1 CD4 cells, and they help activate cytotoxic T cells زي ما حكينا by producing interleukin-2, the same pathway, and initiate اللي هو delayed hypersensitivity response رح نحكي عنه. So the first and third function by the cellular function رح يكون by the th1 cells, and by producing primarily شو رح يكون السيجنال رح تكون obviously interleukin-2 for the first function and gamma interferon for the third function. هلا the humoral response is carried out by th2 CD4 cells. هدول بيحاولوا يدفعوا الجسم نحو a humoral response which help activate B cells by producing primarily, as we know, interleukin-4 and 5. And the role of th1 cells هو gamma interferon also affects B cells to class switch as well. فعشان هذا T-dependent response, it will help the B cells to switch from IgM to other antibodies, IgG and others, and we will see how that works later, that are very more effective in opsonizing bacteria and are more sometimes even more specific to their antigen. فبما أن حكينا في two populations وحكينا كلمة بدفع الجسم معناته there's a sort of a fight between them. They are trying to, each is trying to take the response, the balance towards themselves. فبنرجع the core picture اللي هنا. So this will be a th2 response, or this will be the th1 response اللي هي هذه picture بالمحاضرة الأولى. So again th1 response إذا كانت هاي th1 راح تودينا هون، إذا كانت هاي th2 راح تودينا هون. هاي الاديشن الإضافة بدنا نضيفها هون. So let's look here. So let's, for example, see that a macrophage has ingested a bug. It will produce زي ما حكينا interleukin-12. It will induce interleukin-12 here macrophage. كيف الماكروفاج بتحول TH0 اللي هي النايف helper cell. هاي الهيلبر سيل لا هي مع TH1 ولا هي مع TH2. It is a naive cell. It is capable of being dedicating itself to either of the two populations. هلا أنا طلع عندي a cell macrophage which has ingested this bug and it has خلال مثل pattern recognition molecules has deemed this to be a cellular bug مثلا virus or whatever. It requires cytotoxic response. It will produce interleukin 12. Interleukin 12 will go to the naive helper T cell, which is a helper T cell that is also specific to this virus. فعشان هيك احنا دائما in the normal situation our naive T cells will always become T cells will our naive TH0 cells will always become TH1 cells in the case of intracellular antigens مثلا مثل الviruses. So that's why you will always produce the required cellular pathway, TH1 pathway, for when you recognize this this cellular protein. هلا شو بالنسبة مثلا للانتي بوديز اللي بدك تعملها ضد هذا الريسبونس. So again we don't know who, but we know there's another cell that does the same job. بتروح بتدورها على naive helper T cell and it pushes us toward a humoral response. بتحولها ل TH2 cell. So interleukin 4 induces the TH0 to become TH2 production for interleukin 4 and 5 and then كل الباثوي بنعرفه من هون. So the human cells that produce interleukin 4 احنا ما بنعرفهم has not been identified. فدائما احنا في سجال اذا انا بحاجة more antibody response I would push towards the TH2 response using interleukin 4 and then interleukin 4 again and 5, or interleukin 12 and then 2 راح تعمل لي interleukin 2 the gamma interferon and interleukin 2 for the cytotoxic T cells. So that's what we have here. That's how we push each cell. So can you produce هذا السؤال, can you produce both a TH1 response and a TH2 response at the same time? So the answer would be no. This is a commitment. يعني خلاص هاي الخلية it is always committed towards either population. So you cannot mount a response. ما بتقدر تحول من واحد للآخر. A cell will become permanently committed towards a specific response. بس هل في أكثر من antigen that you can mount a response؟ هذا الجواب الآخر اللي بيحكي لك يعني الجواب الأول كان لا you are committed to either one, but إذا بترفع السؤال بتوسع السؤال شوي بتحكي can I mount a TH1 response and a TH2 response against the same bug? Yes. So السؤال الأول كان can I mount a TH1 response and a TH2 response against the same antigen? No. This is a committed. هذا الانتيجين يا بيعمل لي interleukin 12 or interleukin 4 pathway, TH1 or TH2 pathway. Committed خلاص. بس الفيروس نفسه will have different antigens, right? So you might process different parts and you might mount slightly a TH2 response against a certain antigen and maybe a dominant TH1 response against another antigen. That might happen. So there is a balance between them. TH1 and TH2 cells, for high TH1 TH2 cells. This balance is provided by the production, again, interleukin 12, and increases the number of TH1 cells, cell mediated. So they try to push all these naive, undecided T cells towards a cellular response. فالinterferon كمان from these cells try to inhibit the other side. فشو يعني؟ إذا أنا بشتغل إن أنا ماكروفيج وقاعد بشتغل على th1 side, I will inhibit the other side with interferon, and I will promote my own side with اللي هو the normal stimulatory signal هي interleukin 12. هلا بالعكس الخلايا اللي على ال other side on the th2 response احنا بنعرف stimulatory signal هي interleukin 4. It tries to inhibit the other side. Interleukin 10 is a well documented inhibitory interleukin, and try to inhibit the other side, try to inhibit interleukin or th1 response. So that's a very important here mechanism to see between the balance between the two. So هلا خلينا نشوف comparison. How can we compare? خلينا نشوف معلوماتنا. Can we compare between th1 and th2 cells? Who produces interleukin 2 and gamma interferon? راح يكون interleukin، راح يكون th1 cells. Who produces 4, 5 and 10? راح يكونوا هذول stimulatory، هذا inhibitory for the other side. So this is the th2. Who enhances cell mediated immunity? واحد function 1, and delayed hypersensitivity function 3. So obviously that's the th1 cells. Who enhances the antibody production? راح يكونوا th2. Who is stimulated by interleukin 12, 1, and who is stimulated by interleukin 4? Who th2. So this is another response that we are يعني إذا بتضيف عليه ال T cell maturation. So this is as the cells are maturing, they become double positive. Then you have a single negative CD8 naive cell and a single negative CD4 naive cell. These can become either interleukin 12 Th1 or interleukin interleukin 4 Th2 with the inhibitory. هنا راح يكون interferon راح to inhibit this one, or interleukin 10 to inhibit this one. There's another subpopulation called the Th17 response. هدول هاي موجودة primarily in the gut. This is your gut lymphocyte. خفكر فيها زي هيك. So the TCD8 naive cell is activated by interleukin 27 نحكي عنه بعدين. Tumor growth factor beta. Let's just the T regulatory lymphocyte, which we will mention later on. So that's the paradigm with the maturing. So if you remember this from the first lecture, our antibody or cell antibody mediated B cell will mount our defense against infection by opsonizing bacteria. هاي عرفناها with our antibodies. Neutralize toxins with our antibodies, and viruses with our antibodies. They mediate allergy, hypersensitivity, 1, 2, 3, 4, 5, 1, 2, 3, 4 اللي هم الأربع types of hypersensitivity. And then the autoimmune reaction against your own cells can also be part of your B cell. هلا صرنا نعرف كثير معلومات زيادة هون. هلا حكينا from our cell mediated, so we know now if I want to mount a response against intercellular hosts مثلا bacteria or fungi مثلا tuberculosis viruses or whatever. And then we have more hypersensitivity reaction by so this will be our type 4 hypersensitivity. We have the graft, and هنا this is 1, 2, 3. This is number 4 بنحكي عنه بعدين. Graft and tumor rejection حكينا by how we do that by MHC1, and then regulation of antibody response. So this will be the T cell, the CD4 cell. So now we know more details. So هذا بدخلنا على نحكي راح يكون في محاضرة كاملة عن types of hypersensitivity، وراح يكون أمثلة أكبر، بس بدنا يكون عندنا هلا فكرة. يعني بنحب هذا الكورس إنه دائما يكون عندنا فكرة بعدين نبني عليها لقدام. So الفكرة هنا إنه فكر بالtypes of hypersensitivity is by how immediate they cause disease. How often they cause their sensitivity or they cause their symptoms. type 1 immediate type 4 delayed. تمام. شو اللي عندك؟ تذكر احنا حكينا كان immediate كلهم هدول are اول ثلاثه they use antibodies والرابعه it uses T cells اللي هو بالنهايه راح تعمل لك macrophages which are the actors. So type 1, this is the immediate degranulation of IgE. بنعرف IgE هو بيعمل الحساسيه immediate الانافيلاكسيس الالرجي. ف this is something اول ما الواحد comes to contact with this allergen اللي بيعمل لك يكون IgE mediated وعلى طول واحد بصير يعطس، على طول واحد بصير عنده راش، اذا كان he's anaphylactic على طول بصير معه anaphylaxis. This is immediate. So obviously this is IgE mediated and this is type 1. Type 2 راح يكون antibody mediated. فالantibody is produced over time. هذا antibody binds the cell, causes the cell to be, our target cell causes it to be destroyed, and over time as you destroy more and more cells, target cells, توصل لمرحلة إنه these target cells they fail in their function وبعدين بصير عندك symptoms. هذا هو type 2. So our type 2 is IgM, IgG, direct action على cells بده وقت عبين ما to affect the cells and cause disease. So IgE هذا كان degranulation of mast cells. شو راح يطلع لي؟ هستامين. So الهستامين هون اللي بيعمل المشكلة. IgM, IgG هنا مثلا thrombocytopenia drug induced. So you made these antibodies and then you are destroying your platelets. You're destroying مثلا these acetylcholine receptors myasthenia gravis. Graves' disease you are targeting the thyroid مثلا. Hemolytic anemia of a newborn you are targeting red blood cells. تمام. So all these are you have produced antibodies against a target cell. Type 3 مش زي هيك. Type 3 you have antibodies again, but these antibodies they bind to their antigen, and then you have these immune complexes that are formed, which بصيروا هدول لأنهم بما أنهم كبار they are unable to be يعني they bind to each other and then they start يترسبوا. ففكر بتايب 3 ترسبات. تايب 1 كان ألرجي سريعة حساسية. تايب 2 كان في عندي انتي بيشتغل ضد خلية معينة. تايب 3 ترسبات. ترسبات شو؟ بروتينات. مين هم البروتينات؟ immune complexes. مين هم؟ antibody antigen. So antibody antigen they بصيروا يترسبوا في المناطق عادة اللي فيها البلد فلو بطيء. صح؟ هلا إذا كان البلد فلو سريع راح يمشي هالبروتين معهم. صار البلد فلو بطيء بصير يترسبوا. وين البلد فلو بطيء عادة؟ around the joints, around the fingers, around هذا المناطق. فبي rheumatoid arthritis, SLE, Gaucher, arthritis reaction نحكي عنه بعدين. serum sickness. So كل هذا نحكي عنه لقدام. Can type 3 be quick? You can yes. Type 3 can be quick. بس نحكي عنه بمحاضرته لما نوصل لها. Type 4 اللي هو sensitized th1 cells activate cytokines resulting in macrophage and cytotoxic T cell accumulation اللي هو the normal response, the normal cellular response اللي هو إنك تعمله your th1 response basically. This is also called the delayed hypersensitive reaction because it also can be made against certain allergens. مثلا contact dermatitis إذا مثلا واحد بلبس مثلا copper ring or whatever and then شوي بصير معهم reaction ضد هذا الكوبر. this will be a delayed hypersensitivity reaction بعكس المين اللي هو إذا حط على حالك مثلا إشي that you are immediately hypersensitive to, this will be an IgE mediated. فهاي هيك a quick recap to hypersensitivity so we can move forward. فأنا نرجع للnormal behavior كنا بنحكي عنه. So if you are trying to mount a protective response against a specific microbe that requires either a th1 or a th2 cell, مثلا individual مع TB والTB أنا بحاجة لسيلولر response. هذا الانdividual شو راح سوى؟ صار يعمل th2 response ضد الTB. th2 response antibodies will not really be effective فراح يصير عنده major infection. Humoral immunity is not protected بنعرف لأنه ضد الTB لأنه بنعرف إن TB is a intracellular pathogen. والعكس صحيح، إذا أنت عندك pneumonia streptococcus pneumonia هذه it's a capsulated organism. I need to make antibody against this capsule so I can neutralize this capsule. روح تعمل th1 response, cellular response. It does not infect cells, does not go inside cells. So the th1 response will not be helpful towards the cell. But again, you will have a severe infection. So TB من الأشياء كتير مدروسة and well-known example of how this response is made. فزي ما حكينا it was found that a specific lipoprotein on the surface of that bacterium interacts. Again, now we come back to these pattern recognition molecules, the toll-like receptors, and present on the surface, that is present on the surface of the macrophage, and the macrophage toll-like receptors induces the production of interleukin 12. يعني هاي بتعرف البكتيريا احنا evolutionary بنعرف انه if I produce كيف يعني if I produce interleukin 2 هذا toll-like receptor راح produce interleukin 12 واعمل مثلا th1 response. اذا واحد بني ادم عمل th2 response، اذا كان مثلا متطلع على هذا toll-like receptor and his body does a th2 response، شو راح يصير فيها البني ادم؟ راح يموت and less likely to reproduce هذا البني ادم ويصير عنده اولاد. ف that makes over time بصير كلنا احنا react, we have our toll-like receptors that react towards this pathogen, towards an interleukin 12 producing th1 response. هذا هو this is the evolutionary pressure that moves us towards بصير كلنا احنا نطلع على toll-like receptor هذا بحولنا towards a th1 response مثلا. And the same thing can happen for other molecules for a th2 response. So interleukin 12 نعمل is the stimulating signal, and then this will drive the differentiation of the naive cells to th1 type. Another subpopulation of the CD4 حكينا, they are called the th17. They have been shown to have a significant role in the mucosal immunity of the gastrointestinal tract. حكينا هذولا our gut lymphocytes. They are present from mouth to anus, يعني all throughout the GI tract. And the reason these cells are different because they are producing th17 interleukin 17 instead of مثلا gamma interferon, and they recruit neutrophils to any site of bacterial invasion in the mucosa. And this neutrophils, what do they do? They would come and clear up these bugs. فهم زي ما تحكي are recruiters for neutrophils to clear up infection. So this is the depiction of إنه كيف الخلية بتنقل الماكروفاج بتودينا towards a specific response. So you have mannose binding receptors, LPS receptor, other scavenger receptors, glucan receptor. All of them will tell me to which area am I going to, which interleukin am I going to send, where am I going to try to produce a th1 response or not. That's basically what's going to happen. So let's say the bacteria binding to macrophage receptor initiate the release of the cytokines and these اللي هم الcytokines 70 أنا أودي لحالي, and then again at the same time the macrophage will engulf and digest the bacteria so it can go and present it to the T cells after they are committed to their subpopulation. So how we found the significance of these cells اللي هم تبعين th17 إنه HIV selectively targets th17 response. So بصير إنه دول patients they have high rate of invasion of gut bacteria into their blood. بصير عندهم blood infections caused by gut bacteria مثلا E. coli, Klebsiella, and then they will have these infections. They are also have reduced immunity against fungal infections, which is مثلا called chronic mucocutaneous candidiasis, which is still in مثلا بالesophagus or in the mouth. And again, this is due to the reduction of the interleukin 17 CD4 population. بالنسبة CD8 cells, they are cellular responders, effectors. We know this. They can kill their target cells with perforins, which act as bullets. and then cause holes in these cells. They respond to cytotoxic T cells, مثلا virus infected cell, tumor cell that are producing a virus. فمثلا خليني أسألكم سؤال tumor cell. If you are able بدي أسأل السؤال أعمل شوي pause and then you can pause the video and think about it and listen to the answer. If you can train a CD4 cell or a CD8 cell to produce a response against a mutated protein produced by a tumor cell, will you cure that cancer? فاسمع السؤال مرة تانية وفكر فيها. So that is the basis of اللي هلا صار عندنا بنحكي عنه لقدام كمان شوي بورجيكم صورته and a figure. That is the basis of immune therapy for cancer. One of the bases of the immune therapy for cancer is if you allow your cells to produce CD8 response, cytotoxic response, CD4 activation against CD4 activation مهمة هاي against this cell, you will cure this cell or you at least start fighting it. يعني أقل ما فيها إنه بتقلل حجم الكانسر. You might overcome this entire cancer. So what happens إنه خلايا الكانسر they prevent CD4 cells from activating against themselves. فأنا شو بدي أعمل and how do we stop this بالimmune therapy? We produce an antibody which then activates these inactivated cells. بورجيكم الفيجر اللي بعدين. So the way we kill this is حكينا by perforance. CD8 cells constitute about 35% obviously, عكس 65%. So signature cytokines produced by either of the subsets of the CD4 cells. Your th1 cells will produce gamma interferon to activate macrophages. Interleukin 2 again for CD8 cells. Your th2 cells will produce interleukin 4 and then enhance switching to IgE. th17 will produce interleukin 17 to recruit neutrophils to site of infection. Okay. Activation of T cells. To activate helper T cells, an antigen presenting cell must possess, as we know, MHC2 complex, which carries this antigen that this T cell receptor is reactive to. The activation of the cytotoxic T cell, on the other hand, requires antigen presentation, as we know, on MHC1. ففي من الطرق انك تذكر هالحكي انه دائما لا اذا بتضرب الرقمين في بعض لازم يطلع دائما معك 8. So CD8 times MHC1 equals 8. CD4 times MHC2, 4 times 2 is 8. So APCs both have class 1 and class 2 proteins. واحد من الطلاب سأل إذا في macrophage infected by أو مثلا not producing infected by a virus and not producing MHC1, will a natural killer cell kill it? Yes, it doesn't care if it's a macrophage or anything else. If you're not producing MHC1, I'm going to kill you. So that's one mechanism where we can police the police itself. So this microbe or protein is then is digested by the APC to unique small peptides, and then they are complex with MHC2 proteins and presented on the outside of the cell for the T cell receptor to recognize it. T cells are usually harbored in lymph nodes, and while at the same time the antigen is being encountered عادة بmucosal surfaces where the APCs are present. So how does the APC recruit the T cell from such a distance? An APC اللي لقت antigen and has processed this antigen will start produce a specific receptor on its surface. هذا receptor, it indicates that it is currently presenting an antigen. It has found this antigen and it will start producing this receptor, and this produces chemokine CCR chemokine R7. Chemokine signal for R7 is being produced when? Like a radio wave دائما as being produced in the lymph node. So هيك بتقدر تلاقي حالها بدور على أقرب lymph node. هلا ففكر فيها بهالطريقة إنه أنا APC I'm not presenting anything, فأنا don't put the helmet على راسي اللي هي CCR7 اللي هي بتلاقي السيجنال اللي بتيجي من الليمف نود. هلا كل ما قربت على الليمف نود السيجنال بتكون اكبر لأن الكونسنتريشن اوف ذا كيموكاين سيجنال CCR7 بتكون اعلى. كل ما بعدت بصير الكونسنتريشن يقل لانه بصير له دايلوشن. سو حتى لا يوصل للميوكس ممبرين. هلا انت بالميوكس ممبرين لقيت هذا انت APC لقيت الانتيجين you broke it down you are presenting it you put on this helmet which listens to اللي هي which is reactive to الكيموكاين فبصير يقرب على الكونسنتريشن. كل ما بزيد الكونسنتريشن بصير السيل هاي بتقرب على الليمف نود. That's how it will find its way towards the nearest lymph node. And as for the activation of the CD8 cells, this happens when the APC itself is مثلا هذا حكينا this happens when the APC cell itself is infected with the virus. The virus within the cell is now producing foreign proteins which are presented on the surface of an MHC1 protein and activates the CD8 cell to perform its function. So we know this إنه CD8 cell راح تدور على الخلايا that are infected with this virus and then does its job. So the same happens when a piece of dying infected cell is presented to CD8 cell on MHC1. مثلا also viral antigens are being presented on MHC1. A non-APC will also present viral antigen on the surface on MHC1 as well. And then that's what we just mentioned everywhere. So polymorphism of MHC proteins is needed. زي ما احنا we need a polymorphism the T cell receptor and polymorphism for the antibodies. يعني different types of reactions to different يعني أشكال مختلفة عشان يتعاملوا مع different antigens. So this polymorphism is achieved by having many different genes. زي ما حكينا الفكرة إنه أنا يكون عندي بنعمل with different genes and I take randomly one of each gene pool وبطلع أنا with a different result each time. So MHC1 and MHC proteins can only be present peptides and they are unable to present any other type of molecules زي ما حكينا. And they only present peptides from foreign sources because they will never be reactive towards self-proteins, self-peptides. I think everything here we know. The T cells must be correctly trained in the thymus that we know as to only to produce to not react to our own peptides. Tamam. So that's the same picture that we saw in the beginning. So there's nothing new here. It's just a reminder. So هلا هون دخلنا على the concept of co-stimulation that is required to activate T cells. So everything that we said so far still stands. بس بدنا نضيف شغلة تانية. هلا the first signal هلا إذا ال MHC1 أو MHC2 is binding to the T cell, to the CD4 cell. أنا كتير مهم إذا حكينا if you ever activate a CD4 cell أنت you will undergo clonal proliferation and then you will start activating CD8 cells down the line or B cells down the line. So you have to be absolutely sure before you activate your CD4 cell. So it is important to observe that these T cells they function in stabilizing هذولا زي ما حكينا هو CD4 that stabilizes the connection between the T cell receptor and the MHC protein on the APC. So other proteins then also serve to stabilize the contact between the two, and this will get us towards the co-stimulatory signal. So lymphocyte function associated antigen 2 on CD4 or CD8 cells binds with intracellular adhesion molecules, and that's how the connection is established, and then it is further strengthened by CD4. طيب, let's talk now about the actual second co-stimulatory signal. اللي هي B7 protein on the side of the APC will interact with the CD28 on the side of the helper T cell, the CD4 cell, to proceed with the co-stimulatory signal. إذا أنا مشيت لقيت ال T cell receptor صح وال MHC2 ماسك صح and then I did my lymphocyte function associated antigen, and then I stabilized the signal with CD4. If all this has occurred, then my second signal will proceed اللي هي B7 protein من جهة ال APC و CD28 من جهة ال helper T cell. If you have both, the first signal اللي هي the occupation of the T cell receptor by the antigen كل اللي حكينا قبل شوية plus the co-stimulatory signal, both together will move the cell towards activation. زي ما حكينا once it's activated خلاص بتصير proliferation, memory, and then you can never undo this activation. So if this co-stimulatory signal does not occur, إذا ما عملت co-stimulatory signal, I will not become active. This is a state of energy, and then it will be unresponsive. I will not ever mount a response against this epitope. I will have to use help to reactivate this by producing the co-stimulatory signal. Production of the stimulatory protein, co-stimulatory protein, depends on the activation of toll-like receptors on the APC surface. And هذا زي ما حكينا, this is another mechanism عشان انت إذا at one point you are going to mount a response against self-protein, you still have this co-stimulatory signal which only responds to activation of toll-like receptor. You don't have toll-like receptor against yourself, so that will vastly reduce any future activation after the thymus of CD4 cells against self-proteins. So that's the signal. APC, MHC2, it's B7 protein. This is the antigen. هاي CD4 cell stabilizing the connection. صار اللي هنا كل شيء اللي حكيناه. So now this is our first signal. This is our second signal, which is promoted by toll-like receptors and B7 protein مع CD28. Is activated, بتطلع interleukin 2, activates itself and then activates the CD8 cell. The CD8 cell بتروح بتدور على a virus infected cell that is producing or showing the same antigen, نفسه هذا الانتيجين هون هو نفسه الانتيجين هون, on class 1 MHC2 molecule with a CD8 protein to stabilize the connection and then kill this virus infected cell. فهاي هي قصتنا هلا مع إضافة ال co-stimulatory signal. How do I have polymorphism for the MHC complex protein, the T cell receptor, the B cell receptor? كل الفكرة مرتبطة ب having a pool of genes that I draw from, and then I make different kinds of these molecules. هلا the least polymorphic اللي هي MHC1 اللي هو على كل الخلايا, there's only 3 genes. You either have any of those 3 genes. Matching here will be easier. MHC2 class 2 MHC2, I have more genes. I have about 8 genes that I'm looking at, and they are all present on different sites in the chromosome. So the more polymorphic بالstep الثانية اللي هو T cell receptor. So you have the T cell receptor made out of these 2 subunits, and then each will have to be produced by different genes. So راح نشوف ال T cell receptor actually has a much higher number of genes to draw on from their pool. ال B cell receptor اللي هو راح يكون شو an antibody. فالantibody راح يكون في عنده heavy chain هذا الأخضر and light chain الأصفر راح نحكي عنهم محاضرة الantibody, and each have an antigen binding site, and each part of this will have a pool of genes that I need to draw from. فطبعا هذا راح يكون B cell receptor اللي هو الانتيبادي راح يكون the most polymorphic. راح يكون عندي في أحد institutions that work on antibodies, they are looking at different kinds of different unique antigens وقاعدين بيعملوا لهم library. إلى الآن وصلوا لحديت مليار different antibodies against different antigens, and they're still counting. So this will be the most polymorphic. So now there's to add on this نحكي احنا عن ال HLA هلا. So on the left هون فوق حكينا ال APC presenting to MHC2 T cell receptor specifically recognized. هذا ما في شيء هون ما حكيناه. The co-stimulator signal and so on. So الفوق اللي هو اللي هون on the right is the virus infected cell interleukin 2 produced by the helper T cell and then it is killed. Class 2 MHC proteins consist of 2 polypeptides, both of which are encoded by genes in the HLA focus اللي هم هذولا. And class 1 MHC protein has only one of its polypeptide encoded by HLA locus and a beta 2 microglobulin which is encoded somewhere else. So this is the HLA locus by in one of the molecules by MHC1 complex. Now, if I want to turn off helper T cells, a different protein بدي استخدمه إذا أنا بدي if I want to intentionally cause energy بدي الخلايا ما تشتغل. مثلا أنا عندي مثلا state of autoimmune reaction or something. I want to produce energy. I want to prevent activation. I want to inhibit an activated cell. So if I want to turn off these cells, I will make a signal. Now this co-stimulator signal is no longer, this is a protein called cytotoxic T cell T lymphocyte antigen, CTLA4. CTLA4 is a co-stimulator signal inhibitor, so it inhibits T cell activation. This is a regulatory mechanism to control T cells and turn them off after they have been activated. So بعمل CTLA4 I turn them off. I remove CTLA4, I turn them back on. So here this is what we use CTLA4 in to reduce rejection of organ transplant. And if you have activation, it will be turned off and it will further delay the activation against the graft. So هاي اللي حكيناها قبل شوية. And then this is the other formulation. If this is B7 with CD28, إذا بتحط CTLA4 protein here, this will instead of the CD28, this cell will not be activated. This helper T cell will not be activated, and it will undergo energy. هلا آخر إشي اللي هو so now that we have covered the co-stimulator signal, هذا بنرجع بنحكي عن اللي هو الاميون اللي هو الاميون ثيرابي تبع الكانسر cells. So your normal way is you have this T cell with an antigen receptor and it's waiting for its presenting cell. تيجي presenting cell تعطيها and it co-stimulates it becomes an activated T cell. هاي activated T cell, if it is active against a cancer cell, it will kill it. فشو بتعمل الكانسر cell? Cancer cell can avoid destruction by taking advantage of this co-stimulatory checkpoint and then shut down the T cell using something similar زي ما حكينا لل CTLA4. It will deactivate the T cell. The T cell will not come and bind to it. Even if it binds to it, it will not do anything. فشو الثيرابي اللي احنا بنعمله? We will go and make an antibody that binds this and prevent the cancer cell from inhibiting the T cell. So هذا بصير occupied. هذا يجي تعمل له بحط له antibody هون antibody هون. So these two will not bind to each other, and then this T cell will remain active, and then it will act on the cancer cell, and then these are the drugs هدول اللي استخدموا هم بالimmune therapy. The problem here, I would need an antibody for each different kind of immune checkpoint that each cancer cell is producing. So it's not a pan treatment. It's not something that you can use for all cancers. So من هون بنحكي مثلا agonists of CTLA4 راح يكون inhibitory. هذول بستخدمه مثلا زي بالترانسبلانت مثلا rheumatoid arthritis, autoimmune disorders, which way you can use. Antagonists of CTLA4, things that prevent inactivation. هذول I want to increase immunity مثلا زي ما حكينا as a potential therapy to reduce مثلا tolerance to tumor cells اللي هو هذا الفيجر. So antagonist can be in the form of antibodies. هذا اللي حكينا قبل شوية. Another inhibitory protein of the surface T cell has been described هو programmed th1 PD1, which interacts with PD1 ligand PD1 interacts with PDL1 on the surface of FPCs and dendritic cells, macrophages مثلا, and then will inhibit immune response, inhibit زي CTLA4, and then again antibodies against the system will be effective against and this is a similar story to CTLA4. And we stop here. This is the end of your mid-term lectures. إن شاء الله المحاضرة القادمة تكون part of the final lecture. ويعطيكم العافية.